Hello there, it's Mrs. Scoville, and today we're going to be going through the steps of cellular respiration. This is still in the Unit 5 Flow of Matter and Energy. We have three objectives for today's lesson. The first is to identify the parts of the structure of the mitochondria, to describe the key hallmarks that happen during each step of cellular respiration, and to identify the location of each step in the mitochondria, or cytoplasm, depending on the step. All right, so we're going to start out first with just a little bit of a connection. So cellular respiration, we talked about it at this point, and you've seen a little bit in the lab that we did with bromothymol blue that cellular respiration is definitely affected to your output of CO2. Well, if you think about cellular respiration, your output of CO2 is really an output of your metabolism. And this is because metabolism is the process of energy acquisition, so eating, and conversion, so converting that energy into a form you can use. Well, this is really the job of cellular respiration. So when you think metabolism, think cellular respiration, because the cellular respiration is actually what converts the food energy into energy that we can use. And that energy that we can use, remember, is in the form usually of ATP. There are a few other energy molecules, but ATP is the main one. Something that's kind of interesting is you may have heard that as you get older, your metabolism slows down. This is true because as cellular respiration occurs, it's actually going to put out something called free radicals, and this can damage the mitochondria. As the mitochondria become damaged, they're less efficient, and therefore you don't go through cellular respiration as well, and your metabolism is slower. Because of this, you may have heard a lot about antioxidants. There are certain antioxidants that it's not fully well understood, but people kind of understand that they seem to neutralize the free radicals, and therefore your mitochondria don't become as damaged. All right, so that was just kind of our broader connection. Now we're going to get into the nitty-gritty of cellular respiration. So this is already something that you've seen, so this should be a little bit of a review. But cellular respiration happens in 3 discrete steps, and each makes a different amount of ATP. The first is glycolysis. The other 2 are the Krebs cycle and oxidative phosphorylation. Both the Krebs cycle and oxidative phosphorylation have a secondary name, and basically it goes to 232 with the amount of ATPs made in each step. Also, I've noted here for you, again, this is review, steps 2 and 3. Input of cellular respiration is oxygen, and this is going to be the steps at which we directly require oxygen. Total, this process is going to make 36 ATPs from each molecule of glucose. And just one more time to make sure that you fully remember everything that I'm talking about here, our inputs are glucose and oxygen in cellular respiration, are our reactants. And again, oxygen is what's directly going to be used in the Krebs cycle and oxidative phosphorylation. And we make carbon dioxide as waste, and we also produce water as well. That's not a waste, though. Okay, so I've already told you guys that cellular respiration occurs in the mitochondria, but it's not the whole mitochondria where the process of cellular respiration is going to occur. So we do need to have some kind of idea of the structure here. And the mitochondria, a lot of times we draw them as like little zigzags inside because there are all these folds inside the mitochondria called cristae or cristae. You might see an E on the end, that's just the singular of it. Inside the cristae or cristae, there's an area called the matrix. And no, this is not like the movie. It's just an open area that's kind of open and there's fluid in there. There's also going to be outside of the cristae, but still within the mitochondria, the inner membrane space. And that makes sense. It's inside the membrane, and it's a space. So that's the inner membrane space. And then the mitochondria actually has 2 membranes. So this is kind of strange. It has an inner membrane, which is here you can see folded on the cristae, and then an outer membrane as well. So again, that's just overall the structure of the mitochondria. Later in the lesson, I'm going to tell you guys exactly where each process occurs. But I do want to tell you at this point that only the Krebs cycle and oxidative phosphorylation occur in the mitochondria. Glycolysis does not, and we'll see that in just a minute. All right, we are going to preview the steps of cellular respiration, so we'll go through this short little video now.
Organic compounds, such as glucose, store energy in their arrangements of atoms. These molecules are broken down and their energy extracted in cellular respiration. The first stage of cellular respiration occurs in the cytosol, while the second and third stages occur in mitochondria. In cellular respiration, electrons are transferred from glucose to coenzymes such as NAD+, and finally to oxygen. The energy released by this relocation of electrons is used to make ATP. Carbon dioxide and water are given off as byproducts. Glycolysis is a series of steps in which a glucose molecule is broken down into two molecules of pyruvate. As the chemical bonds in glucose are broken, electrons and hydrogen ions are picked up by NAD+, forming NADH. Glucose is oxidized and NAD+ is reduced. A net output of 2 ATP molecules are also produced in glycolysis for every glucose molecule processed. But most of the energy released by the breakdown of glucose is carried by the electrons attached to NADH.
The pyruvate molecules are modified as they enter the mitochondria, releasing carbon dioxide. The altered molecules enter a series of reactions called the citric acid cycle. More carbon dioxide is released as the citric acid cycle completes the oxidation of glucose. 2 ATPs are formed per glucose, but most of the energy released by the oxidation of glucose is carried by NADH and FADH2. Almost all of the ATP produced by cellular respiration is banked in the final phase, oxidative phosphorylation. The NADH and FADH2 molecules produced in glycolysis and the citric acid cycle donate their electrons to the electron transport chain. At the end of the chain, oxygen exerts a strong pull on the electrons and combines with them and hydrogen ions, protons, to form water. The electron transport chain converts chemical energy of moving electrons to a form that can be used to drive oxidative phosphorylation, which produces about 34 ATP molecules for each glucose molecule consumed.
And that last little bit was incorrect. It is 30. Okay, so now we're going to go through each of the individual cycles and steps. And remember, Krebs cycle is also the same as citric acid cycle, which is what they talked about in the video. All right, so glycolysis. This is going to be the process where glucose is initially processed, and it's split into two. So glucose has 6 carbons. When you split it into 2, that gives you 3 carbons. The name of that molecule, once it's 3 carbons, is called pyruvic acid or pyruvate. And that's just the name of it. This occurs in the cytoplasm of the cell. This will not be in the mitochondria. This process is going to gain 2 ATPs overall. It's a net gain. It's interesting. Glycolysis actually has to have ATP to start it off. So it'll use 2 ATPs to start it. And what this does is it lowers the activation energy of enzymes, and it'll start the reaction. So it's the energy input the enzymes need. But when it actually breaks the glucose into the 2 pyruvates or pyruvic acids, this makes 4 ATPs. So use 2, you gain 4. So overall, it's a gain of 2. This will also make NADH, which is an electron carrier, and it makes 2 molecules of this. In photosynthesis, we have NADPH. That's just a different version of this. Here we have NADH. I do want to point out as well that this is what's called an anaerobic process because this is not going to require any oxygen on its own. So aerobic is like air and ana meaning not. So this does not require oxygen. If we look at this in a diagram, we can see glucose, which again I told you has 6 carbons, input of 2 ATPs. That means you have 2 ADPs that you have left over. It makes 2, and again these are the pyruvates or pyruvic acids here at the end. In the process of these being made, again, this is just going to be a conformational change. You are going to make NADH, which is that electron carrier. This will be used later in cellular respiration, but right now you just need to know that it's made. Okay, so our next one here is the Krebs cycle, which is also known as the citric acid cycle. And each of those pyruvates or pyruvic acids is going to be broken down, and it's broken down into CO2. So this is basically the opposite of the Calvin cycle. If you think about in the Calvin cycle, we took CO2 and made a 3-carbon sugar. Here we're taking a 3-carbon sugar, in this case pyruvic acid, and breaking it down into CO2. This does occur in the mitochondria, and because it is a cycle, it keeps going around. Every time it goes around, it's going to make an ATP, so this will happen twice to make 2 ATPs, and this also is going to use oxygen. So unlike glycolysis, which was anaerobic, this is going to be an aerobic process that uses oxygen. This is going to produce more of those electron carriers for us and more energy molecules. It will make 10 NADHs. So if you remember over here, we had NADH, we had 2 of them. We're going to have 10 more. And we also make 2 molecules of something called FADH2. Just again, another energy carrier. So these are just different names of energy carriers. So Krebs cycle at this point, we're going to break down, release CO2, and we are going to make some ATP. If we look at this in a diagram, it looks a little bit more complicated here, but it's really the same process. So the pyruvates will come in. They're going to join up with this thing called acetyl-CoA. And really what you need to know about that is this is a product that's formed by an enzyme. So this is pyruvate would be up here, and the acetyl-CoA is broken down. This will combine with citric acid. And yeah, that's very much like what you get for drinking orange juice, citric acid. These two will combine together, and this creates a molecule that then can release CO2. So that's what you're actually seeing here. So again, basically this joins together. You get citric acid. This is going to release the CO2, and all the energy, just like before when we talked about ATP, all of the energy that we're releasing here is stored in the bonds. So when we release CO2, we've broken a bond, and that gives us the energy that we need to make NADH, which again is another electron carrier. Each time you can see a bond is broken, we use the energy to make an energy carrier. So anytime a bond is broken, you can use that energy to add to one of these electron carriers. Over here, we're doing some conformational changes, so we have to use that energy actually to make ATP. So again, this is a cycle in which a lot of energy from breaking or rearranging the bonds is being used to make all of these energy capturing molecules. So we have to capture all these molecules so that we can use them to really generate most of the ATP through oxidative phosphorylation, also known as electron transport chain. So this is the electron transport chain, very much like we saw in photosynthesis. The real difference here is what's carrying the electrons and where the energy comes from. So electron transport chain here, we're not going to have any light input. All the energy is going to be coming from those energy carrying molecules, specifically NADH and FADH2. Those were the ones that keep coming up in glycolysis in the Krebs cycle, so they're going to be carrying that energy from the other parts, breaking down glucose, making CO2, and giving us the energy we need to do the electron transport chain. This will happen on membranes inside the mitochondria, and I'll show you guys the exact location in just a couple minutes here. But the main thing you need to know here is that those electrons, the energy stored in the electrons, basically are able to convert ADP into 32 ATPs. The other thing you do need to know, this is the point at which water is released. And we saw that in the little video as well. All right, so here's our electron transport chain. So if you think about the structure of the mitochondria, on this cristae, those little folds everywhere, this is going to be, it's like the cristae membrane where the electron transport chain is occurring. So we've got the matrix inside, and again, just Keanu Reeves is not here, but the matrix is inside, and we're going to have concentration gradient differences just like we saw with the electron transport chain in photosynthesis. And we are still going to have hydrogen ions moving through, setting up concentration gradients so that we can make ATP using ATP synthase. So that's what this is. And if you recall, this is showing an arrow going this way because, as a reminder, ATP synthase, it moves passively. It uses hydrogen ions to move passively, and then it will rotate, and the rotation is what adds an extra P to ADP to make it ATP. Some of you on your test were telling me that a hydrogen gets added. Nope. Hydrogen is just used as the concentration gradient setup. So that's just the energy molecule that moves, and that causes ATP to rotate, and that goes ADP to ATP. Main thing here, though, again, our energy input, there's no sunlight here. So in our cellular respiration electron transport chain, we have NADH and FADH2 being the source of electrons. So in each case, the electrons, just like in photosynthesis, sorry, I keep hitting the thing, the electrons are used to move the hydrogens to create the concentration gradient. So you can see the way the gradient is set up, it's low hydrogen in the matrix, high hydrogen in the intermembrane space between the cristae and the outside of the mitochondria, and this sets up the concentration gradient so you can make the ATPs. That's really the whole goal of this. Use the electrons to power the hydrogens, move them from low to high using active transport, using the energy from the electrons to then make ATP. All right, overall, I do want to point out where these are happening. Electron transport chain, again, I already said, is in the membrane of the cristae. The Krebs cycle happens inside the matrix, so that's what you need to know there with that. And overall, that's it for our lesson. Make sure you go ahead and write your complete summary. And I've also included a link to a video on glycolysis that you can watch in your notes. Please definitely do that. You do need to know all the steps of glycolysis.